So let me introduce our speaker for the day, Dr. Clapperton. Jill Clapperton is the principal scientist and the founder and owner of Rhizoterra Inc, and a well-known international lecturer and advocate for practices that promote soil health. Dr. Clapperton was the rhizosphere ecologist for 16 years at Agriculture and Agri-Food Canada's Lethbridge Research Centre in Lethbridge, Alberta. And then she moved to her family's ranch in the Bitterroot Valley of Western Montana. Dr. Clapperton is also the founder of the Worm Watch program, which uses earthworms as indicators of environmental sustainability and soil health. She has won numerous awards for this work, as well as for her research and education in the area of no-till practices. Dr. Clapperton currently splits her time between the Rhizoterra experimental farm north of Reardon, Washington, and a large-scale regenerative farming project in southwestern Kansas, which I think she's going to talk to us about today. So without further ado, please help me welcome Dr. Jill Clapperton.
Well, hello, everyone. I know this is, wow, this doesn't really have the same feel as everything else, but we're going to try our best. I tell everybody that I'm just glad to see faces, and I'll try and, you know, work with the audience. Now, there are a couple of rules of engagement here, folks. I do want you to interact, and I'm going to ask questions. And I don't know how easy it is for you to answer with the microphone or the way that the setup is in your classroom. So we're going to do our best, but I'll tell you this, that I will stay quiet, and it will get really boring if people don't start answering. So, you know, this has to, we're going to try and make this as fun as we can and do the best that we can under the circumstances. So I will ask you all to try your best to interact. Okay, so, well, let's get started here. So I'm going to screen share. And we're going to start. Now, can everybody see that screen? I need hands up. And I don't mean the other people, I mean the classroom. Yeah, okay, I can see hands. Woo! Loving it. All right. So we're going to talk a little bit about soil health, but we're just going to kind of wander around the place. And, you know, we're going to try and be engaged as we can, given the circumstances. So I thought that what I'd start with, you can see all, oh, well, first you see all my information here. If you have any questions, you're welcome to email me. You can reach me at Twitter. You can also reach me on Instagram. So by all means, ask questions. You can send them to email. That's all fine. If you think of something later, if the classroom thinks of something later, you can certainly email me, and I'm very open to that. I'm going to start with who I am. That little icon on the side was given to me by my son when he was 8 years old. That was 20 years ago. And he wanted everybody who I speak to know who I really am. So that's his mother. He thought that that represented his mother pretty well. And it probably does. Hair always on fire and always thinking about science. I thought because we can't really share much about ourselves, you know, because I'm not there. What we can do is at least you can get to know me just a little bit better. And I hope that as we go along, you'll ask me questions about who I am, how I got here, that kind of thing, you know, if it helps you along your own journey. And at the end, I do challenge myself to understand atomic physics, because if I want to analyze things, if I want to figure out what's in food, how we work on things, and, you know, we have to start understanding physics. So that's just a plug for the physics teacher. It's not the most fun subject for a lot of people, but it's actually pretty necessary. I do hate cooked spinach. It makes me gag. I don't know how many other people that does, but it does me. And I really hate barbed wire gates. I think it's because I'm short. And so barbed wire gates that are strung really tight are really hard for me to deal with. So I'm the first person to get rid of barbed wire gates. Okay, so first off, hands up. How many of you are from a farm? Because I can see your hands. Okay, got a couple.
I'll have to
Oh.
verbally tell you, Milford's got about 4 hands up.
Okay.
Sorry, I'm
Excellent.
still working on the video.
All right, that's fine. I'm happy for people to just shout out if necessary. Okay, livestock farms. Farms with livestock.
About 4 again.
Okay, integrated. Okay, so we've got grazing, we got farming, like farming integrated with it. Yep.
Yep.
Okay, excellent. All right, we're going to talk a little bit about that. I want some of your input on that because we're going to talk about some of the things, and we're going to talk about how soil health integrates with some of our farming challenges. And that comes back to the one thing that mostly I like to grow plants. I grow plants. I believe that I can use plants to build and create soil health, which means I can heal the land, but it also means I can heal my livestock. I work a lot with dairy farms around the world, and you know, dairy cattle, you're always dealing with lactating mothers, which means that they really need high quality feed, and but they need a change in feed. They need a change when they're lactating. They need another change or another quality of feed when they're pregnant. They need another quality of feed when they're dry, and we're thinking about we're about to AI them because we need them to be in their top condition. You need the same thing when you're working on your ranches and your farms. So as we go around, make sure that you're asking me questions about blends of plants or what the plants might be doing. One of the and I really believe this, I think that soil health is in the hands of farmers. This is 2 earthworms. You can see they've been busy here. These are all the cocoons of the egg cases from them being mature adults and having just had sex. So this is the result of all of that. So, this is what we want to see in the soil. We want to break it open. We want to see the earthworm channels. We want to see nice structure. Nothing should be smooth or flat. It should all be really granular. And we should be really having a beautiful soil, smells good, clods up nicely, doesn't break into small, fine pieces right away. It actually breaks into larger chunks, and then we can break it down into smaller chunks. We need to stop erosion. I know, I'm sure you've all heard these messages before, but we need to stop erosion. We need to stop erosion in the Great Lakes. I was just working on that last night. Sediment going into the Great Lakes is probably our biggest concern. It carries with it a lot of nutrients, yes, nutrients and pollutants from various sources, and it actually clouds the water, which changes temperature and lots of other things. So, we need to stop erosion. Wind erosion in the Central Valley of California and various other places like here in Washington State, where we have serious problems with wind erosion. This was actually taken in North Dakota. And these fields were side by side. And if you look at the ditch, you can see the difference in the amount of soil that's blown away. If you think about it, what's blowing in the soil? Is it the heavier soil? Is it the sand? No, it's the organic matter that's blowing away. It's your potential to actually have soil health that's blowing in the wind, or being run off the field in ditches like they are in, we have both. I mean, in southwestern Kansas, I have both wind erosion and water erosion. See the ditches in some of the fields. We need to stop that, and if we're going to actually have healthy soil. So we're going to talk about how plants can help us with that and how livestock also help us with that. So how many of you have seen any kind of demonstrations with the rainfall simulator? Okay. Well, this is one thing I guess we need to get out to ag in the classroom, folks, is the rainfall simulator. And also to our FFA students. This is a rainfall simulator. This is a grazing example here, out of Missouri, actually. This is done by Doug Peterson, who's the NRCS grazing specialist. And what you see here is we have graze, medium grazed, and then really grazed very little. And most people think when they're grazing animals that they can't have erosion. Well, we all know if we get overgrazed, yeah, okay, everything can happen. But this doesn't look seriously overgrazed. But look what happens when we put, you know, an inch of water on there. Look at how much soil has run off in the front bucket. That's runoff. Look what's infiltrated. Not much. Most of it is run off. When we look at the other two, we've got lots of water and infiltration and not much runoff. We want infiltration. We want every drop of water that we have to actually percolate down through the soil and feed those roots and allow us to cool the roots so the plants can keep photosynthesizing, so we can keep making food for our livestock and for our grain crops. So this does the same thing. If we have too much compaction, we have too much bare soil, we have exactly the same result. We have a lot of runoff and not much infiltration. And well, soil is just a big old habitat. When we're standing on our soil, we're standing on the rooftop of another world. And, you know, in our soil are all these creatures, and we're going to talk about that right now. But the point is, as you being all your future farmers of America, is when you build it, they will come. So it's about building the habitat. So what's the first thing that we can do to build a habitat? And you're going to have to find a way to tell me that. And remember that I will stand here and we'll be really bored until somebody answers. So how do we build a habitat? What's one of the first steps that you think of?
Stay up there.
Stay close to
Grass.
how to build a habitat.
Soil.
Say something.
Jacob, you're up there. I was told to unmute it now. How to build a habitat.
How do we build a habitat? What's one step that we could take to make soil a habitat, given that I just talked about erosion? Here, I'll give you a hint.
No, you have to say something.
Water.
Water is a good one. We got something else? Is there something we can do? What can we do to stop erosion? Come on, you farmers out there.
Crops.
Yeah. Okay, we're getting there. How many of you tried no-till?
Or no-till.
Yeah, or no-till. Or how about both? Maybe we could do a combination of both, right? Okay, so let's start building it and they will come. So the top picture is taken in Wyoming, near Cody, Wyoming. It's sugar beet farming, and it's leveling for starting, for preparing for sugar beets. They are all flood irrigated. So they level it and then they ditch it and then they seed. And well, you can see there's been a lot of erosion. It's already super dry. So we have some real problems with the soil biology being homeless here. But then if we look at the bottom slide, we're going straight in with our corn crop into a cover crop. And we're green seeding. So how many of you run the planter or run the disc or sorry, run the drill? I hope you haven't run the disc too often, but run the drill. So why might it be easier to seed into a green crop? What's that?
No one knows here.
Okay, no one knows. Okay. Well, part of it is just because it's easier. When a crop's green, it's easier to cut. It's easier to cut through. It's easier to go through. When it's dry, we can get hairpinning. We can have some other issues, technical issues. So what we want to do is be able to do this green if we can. So trying to get into through cover crops when they're green is a really important trick. And there's many ways to do it. And sometimes it really is just a matter of horsepower. This beauty actually has hydraulics, and we use this in Africa a lot because we're using animals. Something that you may or may not know is that in Africa, men don't necessarily farm. So in a lot of African countries, men are responsible for the livestock. And it's the boys' jobs, herd boys, and their fathers to work the livestock, where the women, generally speaking, will be the ones planting the crops. Because of our programs throughout the world and our food programs, we put the emphasis really on farming, which has been a really big problem in Africa because we've taken the emphasis off of livestock. And not only do livestock, are they important because that's the job of most of the men and the boys in the village, but it's also a measure of your status. So just as you might think having a nice truck or a nice car and some money is your status, in most remote villages in Africa, or even villages in Africa, the number of cattle that you have or animals that you have and the quality of those animals is actually a measure of your status and your wealth in the community. And so women are going to be looking at the number of livestock you have and the quality of your livestock as a measure of how marriable you really are. So that's a completely different way of looking at things from us, but it's really important if you intend to ever work in other countries is to understand their culture and how it works. So I bring animal traction and the use of animals back into the countries I work in. This is Amish equipment. And the idea there is that we can use our animals to use fancy equipment. And there's nothing that isn't modern about that equipment that you see there, except that it's being pulled by horses. Or in the case in Africa, it would be pulled by oxen. But what it does do is allow us to maintain our culture and make sure that we're honoring the skill level of the people that we're working with. So all the time when as you're thinking about ag, not only are you future farmers, but you're also perhaps future researchers. You're also an advocate for food production all over the world. Right now we have to be global. So you need to think about all of that as you're going forward in your lives. Are there any questions about that?
No.
Okay. No? Okay, thank you. All right, once again, we're gonna build this so that they come. Because that's the important thing. And the one thing is that a lot of people go, well, I've never really seen an earthworm in our soils. Well, the good news is as we build up our habitats, they will come. You only need one per acre, and then within 5 to 10 years, you can have quite a number, 200 or 400 per acre. We can make them go faster depending on what we feed them. If we feed them just carbon all the time, like wheat stalks and corn stalks, then they're going to take a long time to reproduce, because that would be like giving you candy for 3 meals a day, which you might like at first and then you might get really sick of. And you wouldn't do very well on it no matter what. So it's the same for earthworms. They need a balanced diet. They need proteins, they need lipids, they need the same things that we do, minerals, all of that in order to grow. The difference is that an earthworm only has to make a specific weight. And once they make that weight, they get to reproduce. So it's all based on the quality of their diet. So if they make the weight really early on in the season, then they can make lots of cocoons. And there can be 3 to 5 offspring in a cocoon. Or if the food is not that good, they can only make maybe 1 cocoon with 3 or 4 babies in it in the season. So it's really up to us. Again, it's in the hands of the farmer. This is a handful of no-till soil. This soil has been, well, first it was run with a noble blade in the early 1930s, 1920s. And then we went to no-till, and it's been no-till ever since. This is a long-term tillage experiment. It's been in no-till or some kind of conservation tillage for 50 years. And you can see that when you take a handful of soil and you really look at it through the microscope, well, it's not that boring anymore. There's lots of things in there, and there are lots of ways to have a look at these things and compare treatments. So to bring things in off your farm, to bring things off other farms, and look at them and compare them. Crystal said that I would be mentioning about my network, JillClapperton.com. We're going to have the network launches next week. We're going to have a space for students to join the network. We're going to give you a free pass to get into the resources for the learning resources. So you can learn how to make apparatus to look at mites. How do you extract them from the soil? To do experiments with earthworms. How do earthworm numbers affect, plant diversity for one thing, plant growth, protein and grain, things like that. We'll have experiments like that. So, if you're interested, then you have my email. Okay, this is close up in person. I said I like looking through the microscope. I really like the electron microscope. It's fabulous. This is the magic of Photoshop, of course, with the colors on the CT. You'll notice that mites don't have eyes. Okay, they're living in the soil, so no need for eyes, don't have a lot of light. But you'll notice that they have quite a few hairs, which we call CT, on them, and actually it's the pattern that we use to identify them into different genera and species. But the pattern is really important because now with nanotechnology, we've been able to place little sensors, and this is magnified 25,000 times its size. So think about, when I say nanosensor, I mean nanosensor on them, and realize that each different species has its own vibration. So how would you tell predator from prey? You can't see. How do you sense them? You go up, pat them on the head and go, hey, are you friendly or not? And if you don't get eaten, I guess they were friendly. But no, they actually sense from the vibration. So the patterns of the hairs actually set up a vibration in the soil. And these animals are actually sensing oncoming as whether they're their own or whether they're a predator. So there are many ways to, make friends and sense your enemies. So this is an example. This is the head of the pin, and this gives you a little idea of the size. So if that's the head of a pin, that's how many of these could fit on the head of a pin. So we're going to focus right now on the biological properties of the soil. And soil health is really those three properties that you see there. Soil health is a combination of both the physical and the chemical properties of the soil, but the biology unites those two. And soil productivity, well, I'm going to talk a little bit about food quality, but not so much today. And hopefully you'll ask me questions about that. Environmental quality, we hear a lot about climate change. Farmers have a really important role with climate change, a really, really, really important role with climate change. And we can talk a little bit about that. I'm sure you're aware of carbon markets. If you're not, write that down as a question and ask me later. And you can see that food quality is really important for health. We know that the quality of the food has some consequences for diabetes, obesity, our ability to fight infections, so immunity, which is really important this time of COVID. You know, so you can see that soil health and the practices the farmers do are directly related to health of the animals and the people. So we're going to start here. These are bacteria, and these are actually chains of bacteria. Each one of these is an individual organism. The crystal that you see here is an endotoxin. This is Bacillus thuringiensis, or BT. You've heard of BT corn, BT potatoes, BT sugar beets. So they've taken the genetic code for this crystal, which is an endotoxin, and placed it in the corn so that if corn borers eat it, they die. This little chain of bacteria, this is magnified 10,000 times its size. This little chain of bacteria, if you just eat one of these, if an insect larvae eats just one of these, it's going to die. That's how toxic and poisonous it is. And plants try and attract these things into the root area or the rhizosphere. I mean, if you're a plant, you're static. You got to sit there, right? Whoa, can't move, can't do anything. You got to defend. So all you're doing all day is just defending things. Or are you? Perhaps you're not just defending. Perhaps you are also being very proactive. And you are sending out signals that attract these kinds of things into your roots and going, hey, you better get on this. I need you bacilli in here because I'm getting a little bit of chewing on my roots. So please get after it. Plants are not static, and we will talk a little bit more about that. Fungi, if you're farming, if you think about food issues, you're always thinking, well, fungi are a bad thing, but fungi are not a bad thing. Probably 90% of the fungi in the soil are all about decomposition. And when a tree falls in the forest, the fungi are the only things that can break down all that lignin. But they need help. Those little guys I showed you before, those mites, yeah. And other things, they actually bore holes in the outside, as do other insects, as do woodpeckers. And they allow the fungi to get in to start breaking down all the lignin and changing that carbon that nobody can use into carbon that's really useful. So all these things are very, very cyclical. So the point is yes, we see some diseases, we have some disease problems, but the majority of these organisms are actually there for our benefit. And when you start seeing this, you know you've kind of arrived. You're breaking down corn stover, and the really hard part, you can see that that's actually a joint, that those fungi are actually breaking that down. And that's going to disappear by the end of the year. That's what we want. We need to recycle the nutrients that we leave in our stocks and recycle it back into the soil. And yes, livestock do help, right? Pounding the stalks into the ground, as long as we don't overuse that privilege. Okay, so this is your first question. What is this? What is this here? And I didn't hear that, so I'm going to need somebody to repeat some of those. Just give a wild guess. And I give marks for being funny, by the way. Always have.
I don't know.
Jacob, what is it?
Bro, I don't know.
Have you ever seen one before?
No, I don't even know what that is.
Okay. This is algae. And algae actually grows on the prairie. In the spring, you'll see them out there. I mean, it looks kind of like a weird looking cow pie, really, or something's had a real issue. But actually, when that dries up, it just goes to powder, and it leaves a huge amount of nitrogen on the surface of the soil. And you do see this in the prairie. You see it in the prairie up north. You see it in the prairie down south. So next time, have a look. It's called nostoc. What about this one? Ideas? And you can take the multiple choice at the top too. Both. Anybody else? It's both. This is actinomycetes. Actinomycetes, although they have their own classification, when you look at them in the microscope, they form chains like hyphae, which makes them very fungi looking, but they're actually in a bacteria category. They're really, really good in soil. They're important for the breakdown. They form like this white powdery coating. Actinomycetes produce a huge amount of antibiotics. Just a huge amount of antibiotics. So they are really important for keeping everything really healthy. So when we see them, we know things are healthy. When you're smelling soil, you actually smell, if it smells sweet, then you're actually smelling the actinomycetes. They have a sweet smell, which is the result of the jasmonic acid that they give off, which they're actually giving off for a reason. It's a calming influence on everything else. It says, well, I'm here and I produce a lot of antibiotics, so everybody just calm down.
So the next time I'm catching strep throat, go out and sniff soil.
That might not be a bad idea. But yeah, I mean, when you're thinking about, and they are related to streptomyces, which means that they're also really good for dealing with potato scab and any kind of tubers that get scabs on them. You actually want to see these actinomyces in the soil because they are direct competitors of the streptomyces, which causes the scabs. So, you know, it's always looking at things. Okay, what about these guys? All right, on the left, what is that hairy looking crazy thing?
Yeah. Bacteria.
Okay,
Yeah, that's our guess over here too.
that's your guess over there too. It's an amoeba. It's a protozoa. And amoebae and all those little hairy looking things are actually like little fingers that help it move through the soil and the water. These guys actually just, you know, it's like horror movies, or, you know, some other movies where we have aliens. It really comes up and it engulfs things. So sort of fingers its way through the soil. And then when it comes across something, it can eat a bacterium or a piece of organic matter, or perhaps even a piece of fungi or something like that. It will actually swallow it, like just sort of give it a great big bear hug and then just engulf it, and move it around till it's actually at its mouth part and then engulf it. So, you know, it's a smothering kind of death. On the other side, what's going on there? What do you see there? Looker's right. And I know we could say that that's a pornographic looking photo too. I get that.
What?
Oh, don't tell me you didn't notice. High school kids always notice that. And you wouldn't be the first ones.
Come on, I was thinking wedding ring, but.
Okay, all right, fair enough. Fair enough.
I don't get it.
Okay, that is a nematode that is being strangled by a fungus. It's been snared by a fungus called a snare fungus. And they form snares and they try and catch nematodes. And once the nematode goes in, it's like a fly trap. It's the movement of it, and then it just just tightens up and tightens up and tightens up until it dies. So they actually start wriggling because they're being strangled to death in this hole. Now, nematodes are also really important for nitrogen. So the key thing here is that between protozoa and nematodes, we actually get 45% of our nitrogen is released by these guys. They're nitrogen concentrators. So if we can let them do their jobs and eat up the bacteria and the fungi and the nematodes and the parasitic nematodes in this case in the soil, then, you know, and recycle them and recycle their nitrogen and really concentrate it around the roots, then we get a lot of free stuff because we let them do their job and our plants benefit from it, and the yield and the nutritional value of our plants benefit from it. So it's about looking at this thing from an entire cycle, thinking about it in terms of how everything interacts. Nematodes, a lot of people are thinking, oh, well, you know, like soybean cyst nematodes. They can be a real problem in some of our cash crops. But then if we build soil health, we start to see more protozoa. We start to see more snare fungi. We start to see that these things aren't a problem anymore. If we can start putting in companion crops and cover crops in, we can actually start rebuilding the populations of the predators. And so in the end, this is about predator-prey relationships. How many have you gone deer hunting? Wasn't it open season on the weekend?
A week or so ago.
Yeah. Okay, a little earlier, and we just opened up where I am. So, do deer have a lot of predators? I don't know. I'm growing a cover crop. I've got 400 acres of cover crops, and I think that the deer are going to get most of it before my cattle do in the spring, honestly. Over 100 head one morning. So predator-prey relationships. We get a lot of bacteria, we get a lot of fungi, they use up all the nitrogen. They use up a lot of the minerals that the plants need, and we need these guys in order to release it. Just like we need more predators to control our deer population so that our mule deer don't get blue tongue, and so we don't start with wasting diseases and things like that. We need predator-prey relationships. It's important, and it's important in the soil too. Here's another good example of a predator. Look at the mouth parts on this guy. Look at this. This is a tiny tank going through the soil, and you see, once again, the pattern of hairs. Hairs on the legs, but not hairs many other places. This guy, just even from the look of it, is going to send off a slightly different vibration. And these here, antennae for sensing vibrations. Recognize this character? I don't know, every time I look at some of the sci-fi movies, especially when they build machines, they seem to look a lot like the critters in the soil. I don't know. I think they've been watching some of these things and taking their inspiration from there. But you can see that this guy is going to go through anything. It's going to eat holes in organic matter, which is going to allow the fungi to get in. They're going to eat each other. It's going to eat through a lot of material. Those are really big chompers. Now, this doesn't look too big. Again, this one is almost 50,000 times its size. So you're seeing something that you probably wouldn't actually see with your naked eye close up and in person. This guy, this is actually an insect. And it does have an eye cluster right there. And it has this kangaroo-like tail, important for decomposition. Anybody got a name for this guy?
I don't know.
Other than kind of cute and fuzzy.
I don't know.
Oh, like a possum.
Okay, cool. This is a columbella or a springtail because they actually hop away. And so you may have seen them when you were in the leaves, like in the fall in the leaves. And you know how in the leaves you'll actually see that everything gets eaten out of the middle and all you get is the structure of the veins left over? These guys have eaten out everything in the middle. They live mostly near the surface and they do blow away. Mites and columbella and these soil microarthropods, they blow away in the wind. And so when we do wind traps, when we put up wind traps, we're catching soil erosion. We actually find mostly organic matter and mites and columbella. And if you have a pretty dead soil, then we just pretty much find the end of your organic matter and clay particles. So again, that blowing in the wind, there's a lot of stuff in there, bacteria, fungi, viruses, all of that. We need to stop it. This is a tiny relative of earthworms. And I would be surprised if anybody guessed that. You can see that it forms like some relatively big cocoons. These are enchytraeids. And enchytraeids are another group of earthworms. They're related to earthworms. There's tiny earthworms that actually, again, concentrate nitrogen in and around the roots. So what you're seeing here is that there's a whole bunch of things in the soil that are actually working to keep plants going. They're working to create a better soil structure so we can infiltrate water, and we can get air, and we can actually get a better ecosystem. I mean, they're all there to help. This is an example. I don't know if anybody's seen earthworm sex before, but this is it. So the term hermaphrodite, what does that mean?
I don't know.
It's kind of like being transgendered.
Yeah.
It means that you're both male and female. And earthworms are male and female. So they carry both sperm and eggs. And so what's going to happen here is the male part, which is near the head, is attached, and right below it actually are the eggs, and they're going to be in, I'm pointing here, but you can't see that. They're going to be in this area here. This is called the clitellum, or the saddle of an earthworm, and when you see it orange and really swollen, it means that they are an adult and that they're sexy. And so the eggs and the sperm are going to go into this little area where they're bound together. They're going to form a foam around each other. So they're very vulnerable at this point to predation. They're going to put a foam around here. And then eventually they're going to back out of this. Once the foam has gelled, they're going to back out, leaving fertilized eggs in a cocoon. And so the cocoon you see is actually the gel casing from this little activity. So something for your trivia. And then this is what it looks like close up and in person. This is a hatchling exiting from an egg. And you can see the little ends here. This is where they've pulled out, and then it's just stretched out a little bit and then closed off at the end. Okay, for all of you studying German, I've translated here. I put slides in foreign languages, mostly because a lot of people feel that if it's done somewhere, the work is done somewhere else, it's better. That's not true. We can do perfectly good work in our own countries, in our own states. It's just that perhaps there's a little bit more cachet when it's done somewhere else. So let's look at this German work. This is plowing. We have mulch, we have direct seed, and we have pasture. And all these colors are different species of earthworms, most of which we have in North America. And you can see that there's not much difference between plowing and mulching, except we change the value of this one here. So we have a few more of the yellow species and not as many of the green ones. And but the percentage of this brown one is kind of the same. These earthworms here are mostly living in the surface, and they grow only for a year. So they cycle annually. Now we get to direct seed, and now we see we have a lot more colors, so we're really building the diversity. What's interesting here is that we have a lot more of this green one. This green one forms big, deep, long, deep burrows. So obviously I lied up in here. There are some deep burrowing ones, but we really get a lot of deep burrowing ones when we get into here, because we're not disturbing the soil at all. So they get a chance to really do things. What's interesting here is as we really don't disturb the soil, we get night crawlers. A lot of you have fished with night crawlers and whatnot, but now we get night crawlers competing with longa, which is a more earth-working earthworm. So now we get two different species of these deep vertical burrowers, but we get a lot more species. So you can see the value not only in diversity of plants, but also in not disturbing the habitat. And I also point this out because it doesn't matter where I go in the world, tillage affects earthworm populations. Where we till, we don't have earthworms. Where we don't till, we have them. And depending on what we grow, we can rebuild the population in anywhere up to 3 years. So how many of you have seen the Soil Your Undies test? Okay, how about students? No? Haven't even done it before? Haven't participated in the hashtag Soil Your Undies? Ah, disappointing. This is a real test, actually. Everybody thinks it isn't, but it actually is. A long time ago in England, when we were trying to figure out decomposition and what was involved in decomposition or how fast decomposition worked, we used to use cotton strips. And you can see that tidy whities are actually mostly cotton, and women's underwear is not. Even if it says it's 100% cotton, that is a lie. There's actually this nylon mesh in there. But you can see that it's still, in where I had a mulch in my orchard, it ate out all of the cotton. Where I didn't have a mulch, nothing really happened. Here we have a cover crop, and here we have the crop. So this is a pretty fun test showing how we can change things in the soil. It's an easy test. You just leave the waistband up so you can actually see it, and then you can pull the underwear out at different times according to your experiment. And
how long did it take to get to that degradation?
In the case of the women's underwear here, that took 2 months.
Wow.
Over here, this took 6 weeks.
A lot faster than I would have thought. Yeah.
It depends. I mean, sometimes people never actually get here, depending on how active your soil is. And it's a pretty nice experiment to actually, you know, take them out at different periods of time and just see how things are breaking down.
How far
patchy
down?
they are.
How far down did you bury them? I mean, how far was it under the soil?
Excuse me. The men's underwear was buried sort of going down in a vertical plane.
Okay.
So this is extra large. So they were going down 6 inches or more. And in this case, I buried them flat at 4 inches.
Okay.
Any other questions? Okay.
I was just gonna unmute to say something real quick,
Sure.
and maybe Dan wants to comment on this as well. We did this experiment, the Soil Your Undies, at our Project Grow demonstration field this summer. And that was just, you know, a fun location to do it, because that is a plot of land that we have been working on through regenerative practices, no-till, and cover crops and grazing and that sort of thing. And it was interesting to see how quickly the underwear were just completely shredded after spending a few weeks in the soil there.
So you can get a rate. I mean, if you sample on a regular basis, you can get a rate of decomposition and know how fast your carbon is cycling. Because remembering that cotton is cellulose. So it's the hardest to break down fibers. So you get an idea of how active your community really is. And it's an easy thing to do. The other thing you can do if you don't want to bury underwear is just bury strips of 100% cotton. And then we have apps now that do image analysis and will give you how much they've degraded. But it's a nice exercise in a really full experiment where you get to graph things, you get to understand rates. It has a nice integrated, from an experimental standpoint and from an educational standpoint, you tick off a lot of boxes on the curriculum.
I've got plenty of underwear left over if any of the schools would like some.
Yeah.
I'd be happy to provide.
Okay, folks, this is another one of these little tests. So I want you all to look at this image right now, like really look at it. And I'm going to tell you what each of these rows represent. Now, these are pop bottles, recycled water bottles. You can do this experiment yourself. You cut off probably the top quarter of the bottle, like where the bottle actually is narrowing down to the neck. Just as it starts to narrow, you cut it off there. And oops. And then you put the neck underneath because you want it actually, and you have holes in the bottom so that it actually drains. What we have here in this one, we have canola residue here. And we have earthworms and canola residue. And the next one, we have earthworms and canola residue, and I had planted corn in there, and you can see that I've got some corn coming up, but canola residue is actually allelopathic to corn, so never ever try planting corn after canola. It's a bad scene. And then this one, we have no earthworms here. Now the earthworms in here, we're at a density of about 70 per square meter. And normally we have up to 200 per square meter. So give you an idea that we didn't have a really high concentration, but you can still see there's some effects. This one is the control where we have canola residue. You can see a canola plant growing there, a volunteer canola. And we got no earthworms. Then we have earthworms and wheat straw. We have earthworms, and in this case, we have CRP. So I went and harvested some CRP residues, and I chopped them up, and I put them on top. And here we have earthworms and cover crop residue. And you can see that there's some safflower left in there. Okay, so what is the first thing that you see? Differences between treatments. What do you notice about the differences between treatments?
Floaties.
There we go.
Yeah, it
Okay, what do you see? Come on. Classrooms.
A plant grew in one of them, I don't know.
Yeah, okay, that's a start. What do you notice? Bare soil? Some of them have more bare soil than others?
Yeah.
Okay, from right to left as you're looking at this image, which one do you think the earthworms like best?
The ones with residue.
Yeah. Okay, I've got a phone going off here. Here we go. Right, but which residue? So this one is this is CRP, this is cover crop, this is wheat residue, and it has earthworms in there, and this is canola residue. So of the residues, which one do you think they like the best? Based on the amount of bare soil that they've buried.
CRP.
Yeah, that's what everybody in the group decided too when I did this demonstration in Indiana. Okay, CRP. Yeah, they liked that, didn't they? This is wheat here. Not so much, huh? What about this one? This one has nothing. What do you notice about this one? This is canola residue, the same as these ones, and this one has no earthworms. What do you notice about these two residues?
Or she's coming out. It freezes on the TV.
The TV freezes and she doesn't.
What do you notice about those two?
Cool junction. Which ones?
These two right here. So can you see my pointer? These two treatments here. If you look across each one, okay, we've got a few plants growing, but what do you notice about the residues? They're both canola. What's the difference between these two that you see just immediately? Don't think about it too much.
There's less than one than the other.
Yep, that's true.
Well, not composed.
What about the color? If these are the same residues, look at the color there and look at the color here.
One looks darker. Darker.
Yeah, darker. So this one has a lot of bacteria and fungi on it because it has no earthworms. So look what the earthworms do. They actually come out of the soil at night and they suck off all the fungi and the bacteria off the residues. So think about that. What do you think that would do if I was struggling with plant diseases in my field? Why do I need earthworms? I can hear people whispering.
What?
The worms will get rid of it.
Yeah, that's exactly right. The worms actually eat up fungal diseases. They actually prevent them. So by burying the residues and by actually wanting to eat fungi, so do the columbella. The columbella in the soil actually prefer to eat fungi. They prefer that because the fungi are concentrating nutrients in their bodies. So they're a way better nutrient source than actually trying to eat the organic matter itself. So what they're doing is they're actually going to eat what's eating something else, so they eat a more concentrated food source. So we can actually manage fungal diseases by also building the soil health. So characterizing a healthy soil: good soil structure, predator-prey relationships. We need pore spaces, because the only thing that actually actively burrows that I showed you were earthworms. Earthworms are the only ones that actively burrow. The rest of them can't really burrow very well. So they need the plant roots and the bacteria and the fungi to make soil structure so that they can move around on the water films. We can't have predator-prey relationships. The amoebae can't get at any of the bacteria and the fungi if we don't make spaces for them to move around in. So just like a city, they need they need some kind of infrastructure. An idea, look at that plow pan there. We've got some kind of soil structure and then this just sticks out. But am I worried about that? Would I put a plow through it? Would I do any kind of vertical tillage? No, because the roots have actually gone through it. So I'm gonna let that work. This soil here has been no-till for 18 years, and you can still see that line right there, which was the plow pan, but it's really disintegrated. This is a really easy test. put a spade in the ground, take it up, and then have a really good whack at it on the ground to sort of loosen everything up, and you will see things will just, you'll see the plow pans perfectly. So when we were looking at for like soil structure, one of the best ways to look at soil structure is actually to take cores, like you would for bulk density, soil bulk density, and put them in a CAT scan or do an MRI. And when you do CAT scan, you get things like this, where you can actually see fungal hyphae, you can see bacteria, you can see the spaces in between there, and you can see that this looks like a road map, right? Like, pretty cool. This is what all those animals I showed you are moving around in. This is their world. This is also their world. You can see that their world is actually filled with water. So there's water films on all these particles, and they are zooming around in here. This is like a major waterway. This is the Mississippi or the Missouri here. And then these are all the little tributaries. And they're zooming around in here, consuming bacteria and fungi. Bacteria and fungi are finding things to eat themselves. And we have this whole little world. This here is actually columbulan poop. And when the surface animals like columbula, that little furry guy, the possum guy, they actually create their own soil structure, and so do the mites. So they contribute to building a lighter, fluffier, more tillable soil, if you want to call it that. So they're actually also contributing. But the other thing they're contributing is that little round packet of organic matter is also a place for the bacteria and the fungi to go to get nutrition. So like you're starting to see, we start building soil biological activity all the time. We're always building it. And roots can access more things when they do. So this plant that's living in poorly structured soil can only have a few roots, because what is it spending? Spending all its energy trying to move through the soil. So plants actually expend energy just like you do. So this would be like you trying to swim in mud. It's not easy, and you're going to expend a lot of energy. This would be like you swimming in, you know, let's call it a slough, but in a pond for sure, with a little bit of sediment. But you're going to be able to swim quite easily. You're not going to expend a lot of energy. So you can spend more energy on above ground, and you can spend more energy growing more roots. The other thing is that because you have excess energy, you can now go exploring a little bit, and that means that you can produce more lateral roots, which means that you have access to more nutrients, which means again that you can grab more top growth. And you're probably getting more nutrients because there's a whole lot more predator-prey relationships going on here, which means more nutrients again, and off we go. This guy here is going to struggle, and you can see topped out at the plow pan, and so now roots are going lateral, and it's trying to find food, but you can see it's clearly not. There's a lot we can do when we start having good soil structure. We can start growing plants, and it's all in your hands. As the future farmers, you can do this. You can decide that you want to have healthy plants or not. It's up to you. And that's what soil looks like. Now, once again, I'm going to let this guy go here. It's going through that platy soil. So I'm going to try and use plants. And I said that I like plants. I'm going to try and grow plants that have big roots, big, deep tap roots that are going to bust up some of this, so I start getting closer to this on the right-hand side. So how do we create healthy soils? Because we can create them. Once again, this is all in your hands. So what do we got here? Other than downright amazing. I'm sure somebody recognizes that yellow flower. I think we're muted.
Called a sunflower.
Right, sunflowers. And what do we got here? What's happening in this field? Like, what kind of agriculture is this? What do we got there?
Those are flowers.
Yeah, we have sunflowers. That's the cash crop.
It's like a companion crop of some kind, but
Yeah, that's right. We have a companion crop that actually turned out to be an intercrop. It's faba beans. So this is faba beans, and this is crimson clover. And originally, this was a companion crop to feed the sunflowers, which you can see are doing really well. And this is on dry land agriculture in a 9-inch rainfall zone. And you can see here that we've got a really nice crop. What turned out is that we took sunflowers and then we also took a crop of faba beans afterwards. So it was a twofer. Faba beans are considered to be a superfood. So they make a lot of the protein powders and whatnot out of faba beans. So it's a really nice market for both of these crops. These went to Spitz for confectionery. These went into the paste market, the protein paste market. But what have we done here? We have covered the soil completely. Got weeds? Think not. Can we spray? No, we can't. That's the whole idea at this point. We're trying to minimize the amount of herbicides and things that we need. That was the idea here. It turned out way better than Josh thought, but worked really well. And we started to see things like this. This is a carabid beetle, and this was a slug. Predatory beetles. We talked about what goes on inside the soil, but what goes on the top of the soil. When we start using a lot of insecticides, and sometimes we have to use them, but when we start using them a lot, we stop this happening. And then we get into other problems. Think about the fact when we get really bad infestations of aphids. Well, if we can leave, like on those sunflowers, I can get aphids. If I let the aphids come and I can wait it out for 3 to 4 days, which I usually can, I start to see all the ladybugs go into full-on munching mode. And the ladybugs themselves will be eating it, and their larvae will eating it, because it's the prey comes first, the predators come second. So we have to allow for that lag time. And sometimes we're impatient, or we are doing prophylactic spraying and control without allowing for natural cycles. So it's important to make an informed decision based on science. If I have swept, if I've used my sweep net out in my field and I am seeing lots of ladybugs, then why would I spray? Because I know the next thing that's going to happen is they're going to eat all my aphids. So that's the kind of thing that we need to think about when we're doing some of this work. Same on canola fields. It's like if I wait a little bit, I'll get control. And I'm always looking at, well, what is the economic damage? What do I need to really decide to do? Do I need it? Do I not? Margins on crops, economic margins on crops right now are not very good. So I'm going to try and lower my cost of production as best I can and let nature do her job while I do mine. And part of it is doing this. You look at these animals in here, and we're growing cover crops for them. We are improving pastures. We are seeing the difference. You can see the difference in the background with the non-green and the green. This isn't under irrigation. This is the native grassland, and this is what we're growing here, because we're growing things in the off season. So we end up with green in the early season, and then this will green up later. And it's pretty much just like the tall grass prairie that you all know as well. This is, and we can do this. We start doing this kind of grazing. We start growing this kind of quality forage, and we're going to put 3 to 4 pounds a day on these animals. And I'm not going to explain that because you're all in FFA. You should know what that means. Above ground diversity is a mirror to below ground. There's no reason why we, you know, when you saw the sunflowers with the faba beans and the crimson clover, I got 3 crops into one field at one time. I just increased my diversity. If I can do that with every crop that I grow, every cropping season, I'm actually increasing my diversity. I am creating pollinators. I am changing the soil structure, and I'm building the soil structure. This is an example of all the different bacteria that just in one drop of water, all I did was take a bit of soil, I added water, I swirled it, took a drop out, and did this test. This is what the soil microbiometer is measuring, which is a really nice little test that you can do in the classroom. And microbiometer, Dr. Judy Fitzpatrick, who developed that test, she also is the inventor of the first HIV home test. She developed this test, you know, that everybody can use to look at their soil microbiology and get a reading. So if you're going to do experiments like that, I recommend that you check that out. Okay, we have to have some data, and I put this in because I'll tell you what PLFA mean, and PLFA is this: phospholipid fatty acids. It's a way that we can look at the living microbial biomass, in this case, the living bacteria in the soil. They're living because we are measuring the phospholipids, which make up living membranes. And when things die, including us, the phospholipid membranes decline really quickly. So we know that when we're measuring phospholipids that we're actually measuring living biomass. So these are fallow. I don't need to tell you that this is wheat fallow. This is a mustard cover crop. This is a mix of 4 species. And this is a patented mix called TerraLife. So tell me what's going on in this graph. And this is the sampling date. So this is how much microbial biomass, bacterial biomass there was with each one of these crops. So maybe let's just start with the fallow and what happened with the fallow, because this little first sampling date is kind of interesting.
The bacteria rate went up.
Yep, exactly. So where we had plants growing, we had more bacterial biomass at 28 days than we did when we had a fallow, right? And then what was interesting is TerraLife is a mix of about 8 to 10 species. So what do we see here is that after that, as we took more time, the microbial biomass, bacterial biomass actually decreased with these ones, with these treatments. And up here, we actually maintained a higher microbial biomass, which means that we had more bacterial, like more predator-prey interactions. So I know graphs, when you see them in your presentations, they always kind of look boring, but it's important to pay attention to them sometimes. And you just, again, don't overthink it. What is your first impression? So this is a really good example of a rhizosphere in a cover crop. This is TerraLife. This is a cover crop that I worked with at DSV Seed out of Germany. Look at that. That's a rhizosphere. this is the way we might look at a rhizosphere. People of the First Nations, this is what they would consider a sacred circle. And so we have the plants. The plants really drive the rhizosphere. So every different plant you put in the ground will create its own rhizosphere, its own group of microbial community around its roots. And that, the plant roots themselves, and combined with the organisms, will actually change the physical and the chemical structure of the soil. And you've seen this. Dig in a soybean field. It's easy. Dig in a cornfield. Not so easy. That's the effect of the roots themselves, the plants. So this is everything, this is a sort of an idea of what's going on in the soil. Roots are leaking. Symbiosis. Can anybody give me an example of a symbiont? I just talked about soybeans. There's a hint. Nitrogen. Second hint. So what helps soybeans?
So the nitrogen fixing bacteria.
Correct. Right. They're symbionts. Soybeans actually need another symbiont called mycorrhizal fungi, which help them get iron and zinc and phosphorus so they can actually form the nodule. And we have nutrients going in and out because plants actually control the flow of nutrients. So if they need to get something, they'll actually change the ionic balance in and around their roots so that they can attract other minerals. They will send water in and out because in a really dry time, if they need the soil to actually bind to the roots, then they'll actually lose some of their own water in order to bind the soil to the roots. We don't ever want them to do that, but they will do it. And there's all these things in the soil. Some of them are promoting growth and some of them are inhibiting growth. Most of your pathogens are trying to slow the plant root down. And most of your beneficial bacteria and fungi are trying to speed it up. They're trying to speed it up because they want more roots. They need more habitat. The pathogens are slowing it down because they need to jump on because they're slow and they're not good at competing. This is mycorrhizal fungi. So here we got the root. These are the hyphae, so the strands of the fungus itself. And you can see we've washed away a lot of things except the sand particles, which are in there, and some of the clay. And you can see how the fungal hyphae are actually binding all the soil together. Again, trying to create structure. This is what it looks like. This is if we don't have the mycorrhiza attached to the roots, because they're a symbiont, they actually grow inside the root. They can't grow without a host. So if you're a Trekkie, they're like a trill. They actually have to grow with a host. And the host actually benefits from them. So the mycorrhiza provide nutrients and expanded root area to the host. The hosts have to provide the carbon because these organisms, the mycorrhizal fungi, can't produce their own carbon. So they rely on photosynthesis for that. And not just sugars, because everybody, when you say carbon, thinks of sugars. That means amino acids. It means organic acids. It means lipids. It means complicated molecules that have carbon backbones. So now, see how important your chemistry is starting to become? So mycorrhizae actually mine nutrients from old organic matter too. So what I want you to notice is if you look here, just across these two roots, what do you notice about color? And I want you to use all your senses. You can't smell this, but I want you to really look. Is there a color difference? Do you see a color difference?
One's lighter.
Yeah.
Yes.
Yeah.
Yellow.
Yeah, yellow. Exactly. Okay. The one thing is yeah, all of that. So what you have here is you can see the nodules here. This is a legume. And it's actually it's been mining the organic matter from this old rootstock. These are mycorrhiza. When you see the yellow color, it's actually mycorrhizal fungi. It's the only way to see them without a microscope or staining. And I'm going to go past this. We already talked about this a little bit. Oh, darn. Thought I had that. This yellow color is really important. It's a way we can tell mycorrhizal fungi when they're colonizing the roots. So we know that we actually have some good soil health because we're seeing yellow roots. We're going to show you how to do that more. So they trade minerals. Mycorrhizal fungi are really important. They don't like tillage much either. They are important for creating soil structure. They actually increase your aggregate stability. And you can see that when we get into no-till, this is plowed, this is no-till for 1 year, 2 years, 3 years, and then this is a pasture. You can see the difference. This is the effects of plants and our management in creating soil structure. And again, remember, soil structure is one of the first indicators of good soil health. And here we go. This is a fluorescent stain from Chris Nichols, and you can see here's our root, here are the hyphae, and these are actually spores. So these are the reproductive structures of the fungi. And now you can see it holding everything together. Here's some of the roots, and then these are all being held together. This soil is being held together by mycorrhizae and glomalin, the glue. This is a really bad weed. This is knapweed. Knapweed is highly mycorrhizal. So this is the structure called arbuscules where the plant and the fungus actually trade. So this is the original carbon trading. This is carbon trading. That's what we're doing. We're carbon trading here. The plants don't stop mycorrhizal colonization. They see it as a friend. So no plants actually ban it. They actually allow it for whatever reason. They recognize that this is not a pathogen and they allow for the colonization. This is an example again of that yellow. See the yellow here? These are onion roots. Onion roots are great for baiting mycorrhiza. You can see it here and a little bit here too. And I have a method where you can actually put these roots out on a graph paper and then actually get a percentage. And it's very close to the actual percentage when we stain and doing my microscopic assessment. But baiting plants, all plants that form mycorrhizal fungi, form mycorrhizal associations easily, will form yellow color. And there it is there. You can actually see it. This is the control, but we still have a bit of yellow color. Not to the same extent. The plants form the yellow color. We don't know why, but they do. Here are your plants that really require mycorrhiza. Corn is one of them. You're in corn bean country. Wheat country as well. Wheat, not so much, but corn and beans, yes. And all these other plants as well. These guys don't need it as much. We put these canola and mustards and whatnot in there. Doesn't matter that they don't form mycorrhiza. We can make a mycorrhizal mix in our cover crops and whatnot. I would still put these in because it's still a balance. Because these guys, these brassicas and crucifers, they actually feed our microarthropods. They love it. It's like Red Bull for them. They go crazy on it. And so we need to have a balance because we need predators, we need prey, we need the mycorrhizas. So we're going to try and balance our cover crops. So when you choose a mix that you're going to see it as a cover crop or as a companion crop, you want to be thinking about all these properties that these plants have. So here's a faba bean, a soybean, and here's your corn here. This is the pH of the root exudates. Every different plant attracts its own microbial community. See, faba beans are going to attract things like a lower pH. Soybeans are going to sort of hit the middle ground, and corn is going to hit a more neutral pH. All of that is going to affect the microbial community that lives in and around those roots. They are active. Plants are not static. They are trying to make decisions about what grows on their roots. Are these mycorrhizal? Off mute.
What was the question? Oh.
Do you actually see mycorrhiza here? In this picture? Can you tell if these roots are mycorrhizal?
Yes.
So how do you know?
They're yellow.
Right, you can see yellow ones. So you can see the yellow here. So plant interactions. Really, really important. Creating plant communities is the key, and roots are connected. The mycorrhiza will network. So we know from experiments that we've done with forest trees that you can label one tree with radioactive carbon in a forest, and that carbon will travel through the mycorrhizal network anywhere up to 25 kilometers. That's a really long way for a carbon molecule to go. So remember that the mycorrhiza are networking our plants. They're conveying disease resistance, drought resistance, because they're moving water between them too. This is flax and chickpeas growing together. Again, this is something that we need to spend more attention and really start working on, is what things grow together. These two crops, when they grow together, do something called out-yielding, which means that they yield more together than they do separately. So even though I only have half a field of chickpeas and half a field of flax, I get more than if I had a full field of flax and a full field of chickpeas, because they like each other. Wheat and chickpeas will do exactly the same thing. You put chickpeas between the rows of wheat, and wheat will easily hit 15% protein, and it will grow way better than it would by itself. Feeding, because we actually have active feeding happening here. These chickpeas are feeding nitrogen. These ones are stabilizing the soil. They are sharing together. And look, do you see much soil underneath there? You can't see it. These two are two crops that are not very competitive, but grown together, we don't get weeds. And we don't get diseases, because flax doesn't host any diseases for chickpeas. Chickpeas get a little bit of a rest. They don't get the same wind. There's a lot of advantage to growing things together. In healthy soils, plants thrive. We detox things. We don't have erosion. I mean, there's a lot of good reasons. We increase soil services. So what's happening here? What is this?
Dung beetles?
Yeah, it is, dung beetles. It's dung beetles, and I'm sorry that I didn't put something beside it to show you how big that ball really is, because it's the size of a quarter. It's pretty amazing what they can do. Yeah, this is a dairy pasture. But the point here is that nature doesn't grow in straight lines. You don't see straight lines in nature. You see like a plow layer, a compaction layer, it's a straight line. Nature doesn't work in straight lines, perfect spheres. These are all scanning electron microscope images of various mites, some of which, like this one, you wouldn't even think was a living organism, but it is actually a mite, and it does move. So with that, I am done, but I'm hoping that you guys have questions for me. And not to shame you or anything, but when I talk to kindergarten classes, which actually get a very similar presentation to this, they have lots of questions and lots of stories, which I generally don't get from high school students, and I don't get from my university students at all until we get going after a while. So I'm hoping that you're going to hit the in-between ground and you've got some questions, even if they're more about me or what I do, or, you know, university, what classes, things like that. yeah, I hope you have some questions about this. So we'll unmute and just make it a free-for-all. Unmuting. There we go.
Hey, who has questions, you guys?
So, you said in Africa, like, men don't really work on, like, planting and stuff. Do a lot of countries, or a lot of countries like that?
Yeah, I did. Yeah. So
He asked if
men,
that's or another. Yeah.
so men are men. A lot of the status of a man in most of the communities is related to the quality and the number of their animals. I didn't realize this at first, so I'll tell you a story about that. I did not realize this at first when I first was working in Lesotho, which is a small country in the middle of South Africa. And so I was working in Lesotho, and I was working in the field with, there was a few men and there were a few women. And this one man came up to me and he was working beside me, and we don't work with anything but hand tools. So you kind of practice your golf swing while you're killing weeds. And what I would have done for a weed whacker, I can tell you. But gas, anything with fuel meant that a woman had to carry it on her head for 10 miles. And, you know, it seemed like a small price to pay was like practicing a golf swing or using some energy or getting fitter rather than carrying 10 gallons of fuel on my head. Yeah, just seemed like a better choice. Anyways, this man is next to me and he comes up and he starts telling me about his cattle. And I'm going, yeah, like, oh, wow, okay. You have like, he was telling me about how good they are and, you know, how he's got his farm and he's got a few pigs and he's really building this up. And I said, well, that's really wonderful. And then he starts asking me, he says, well, how many animals does your husband have? Well, okay. I don't want to tell him that I have, you know, 200 cows in the field like right now, because that's like, he's got 3 and that's a big deal. And, you know, 200 cattle doesn't make me really wealthy either in by our standards, right? So, so I'm like, well, you know, has a few. Well, you know, what is the quality of them? So I said, well, you know, they're pretty good. You know, he had twins and some things like that. So we chat about it and he goes on about it and he goes, I would like to make you my first wife. And I was like, oh, well, thank you so much. He said, you know how many cattle I have. I'm sure that my cattle are much better than your husband's cattle, your current husband's cattle. And you would be my number 1 wife. He had 3 wives at that point. And I said, no, well, you know, I'm okay with that. No, I just need 1 husband. That's enough. And we went on from there. And then I had to really learn after that, you know, so then I went back to the village and sat and talked to the people. And I said, I need you to explain this to me. And the more I started working in Africa, I realized that our programs, a lot of them, we take out the animals because, you know, it's like, well, you got to have food, but you also have to have status. All of us want to be wanted, and all of us want to be relevant and feel that we are relevant in our lives and relevant in our communities. And when people don't feel like they're relevant, or they don't have a role, or they have no status, or they have no stature in their community, they tend to turn to drugs and alcohol pretty quickly. And that's really not good for women and children. So one of the things I do is I really spend a lot of time with the African Conservation Agriculture Network, working on animal traction, which has brought me a little bit of flack because everybody wants to, you know, go, well, you need to have tractors and you need to have power tools, and, you know, it's about becoming modern. But people there walk almost everywhere. I mean, I worked in all the poorest countries in Africa, and people walk everywhere. People don't drive, not even with motorbikes. They ride bikes. They don't really do a lot of mechanical things, so they don't really understand combustion engines very well. And fuel stations are few and far between, and when you're out in the farms, fuel stations are really far away. So if you are in a small village, you are walking a long way with fuel. So you're carrying fuel. You're either carrying fuel with a cart, with a donkey, or you're getting somebody to transport it, or you are transporting it yourself. In most cases, it's women transporting it on their heads. So you'll see them walking with fuel cans on their heads. I look at that and go, well, I think that something walking itself is probably easier right now than, you know, walking around with fuel on your head. So, and the other thing you see is you see tractors out in the middle of the field, not with nothing happening to them because you can't get parts, don't know how to fix it. There's not like somebody you can call to put a tire on. You know, like I punctured a tire on an antler, you know, while I was seeding my cover crops, and, you know, I couldn't change a tire on my tractor. So I called somebody and they come out and change my tire. Well, if I'm in Africa, that's not happening. So I'm leaving that in the field until I can round up, you know, 10 people and try and sort it out and try and get that tire off and try and fix it. So, you know, the point I think I was making there is that you really have to work with the culture of the people and really understand their culture if, you know, you're going to be helpful and you're going to help them, because in the end, it's about facilitating their ability to grow food. Not your ability to feel good about yourself and that you're bringing modernization to them. If they don't want it, then it's about taking baby steps. It's the same as you in, you know, trying to convey your own cool ideas about how you might change things at home to your parents and your grandparents. Baby steps, one step at a time.
Thank you.
So, anybody else?
No.
I really want a Kit Kat.
Go ahead.
Hold up, let me get
How often are you out of the United States, like in other countries?
Used to be a lot more. And I decided that I was working a lot in poor countries, helping people grow, build their soils up and grow nutrient-dense food, because it's important that not only do we grow more food, but we need to grow quality food. Because if you don't have access to a lot of food, right, then you need to have the food that you eat needs to be really nutrient dense. So you don't need to eat so much of it to get your nutrients. I was working a lot, so I was probably gone every month somewhere, sort of home for 2 weeks, gone for 2 weeks. So my son spent a lot of time out of school. He used to be out of school, which, you know, teachers close your ears. He used to be out of school for anywhere from 40 to 60 days a year with me traveling and doing his homework somewhere else in the world. So he pretty got used to this COVID kind of distance learning pretty early on in his life. So we were gone, you know, quite often. But then I realized one time I was on a Navajo reservation in Arizona, and I realized that the conditions there were just as bad as anything I'd seen in Africa. And it was time to be home and work at home and work for the people that are around me in my communities. Not that it's not important to be global, but it was just as important to be at home and see if I could make changes at home. So I started working more at home. I still have projects. I still work in the EU. I have projects in in a number of European countries right now. Again, we're using companion crops, doing integrated livestock grazing, high density stock grazing, introducing that, rebuilding pastures, like just rebuilding pastures with plants. And starting to, increase dairy production. I mean, and most of my dairies, we've doubled the production, and we've just spent more money on seed and a no-till drill. That's it. And we're up on milk production. We can feed twice as many cattle on the same number of acres. Can be done.
I got a question.
Yeah.
What is your most favorite place that you have visited to go help out?
Oh, I love South Africa. Love South Africa. I love the diversity, from the Western Cape up to Namibia. Yeah, it's, going through the Kalahari Desert, the elephants in Addo National Park and Kruger National Park. Being close to elephants is just pretty amazing. They are stunning. Watching a cheetah stalk a gazelle, it's like you're in a fairyland. And you have all this diversity of landscape as well. So the plains, the coasts, the interior, it's phenomenal. I wish it weren't so dangerous. I actually have a business in South Africa. I'm partnered with my business partner there, and who's a South African. And, it's just, that's the disappointing thing, is that it's just a bit dangerous. But it's spectacular. The country itself is absolutely amazing. I mean, I love Australia too, but yeah, my favorite, so far, I have to say that I love South Africa. I would move there probably if it weren't quite so dangerous.
Was there ever a country that you had trouble researching in?
Sorry, country that I had trouble what in? I need you to repeat the question.
Was there ever a country that you had difficulty researching in?
No, actually, I mean, I like that question. No, there hasn't been a country that I've had difficulty researching in. I have been very fortunate that everywhere I've gone, people have welcomed me, really welcomed me, and welcomed me into their villages, welcomed me into their homes, into their research facilities. Yeah, I've been very blessed that way, very blessed. I haven't had, in any of the countries I've been in, nobody has sort of said, no, you can't come in. It was a bit, I mean, Russia was a little interesting. Just getting, they didn't stop me, but when you go into the country, same as Ukraine, there's a lot of scrutiny, and you really feel like you're in a police state when you come in. I mean, you're out on the tarmac in the airplane, you're escorted in, and very stern looking immigration officers are questioning you. And most of them, even if they do understand English, just plain out won't understand English. So you're kind of pointing and trying to, give some idea of what you're doing. Usually when I'm in those countries, I'm with a translator. But not until I get through immigration. So sometimes they're yelling at me on the other side to, what somebody's asking you. Yeah, that can be, it's kind of interesting. One time I got delayed in Kyiv, in Ukraine. And I was staying in what was supposed to be a hotel near the airport. And Ukraine is not a communist country, but I mean, it's been that way for I mean, it's been under communist rule, so it has a very different feel. And I got to the hotel, checked in. I mean, it was wild. You just couldn't believe that anybody wasn't scamming you right from the beginning. And there were monitors at the hallway, at the end of the hallway in the hotel. And even though it was like a 4-star hotel, I mean, you might as well be sleeping in a dorm room. So it was kind of an interesting experience. So I've had some pretty interesting experiences in those travels and learning about the countries. But nobody's ever stopped me from doing research. They've always welcomed, yeah, I've always been welcomed in.
Go ahead. Is there a place you want to research that you haven't yet?
Yeah, I haven't really had any time to be in, spent much time in Asia. And I'd like to spend some time in Asia learning. And that means even like, you know, India, Thailand. And I will be spending probably a little bit more time in China in the future. But yeah, I think I don't actually in Mongolia, but I think it'd be really interesting. I like traveling. I like learning about culture. The funny thing is that people ask me, well, did you go to the, you know, they ask me about being in the city, and I rarely ever get to be in the cities. I'm always out on the farms. So I always feel that I get a real pulse on the people of the countries I'm in because I'm with the rural people, the real people. So yeah, I'd like to spend a little bit more time in Asia.
Yell it.
What made you want to become a soil scientist?
I guess I was actually mentored, and you'll find this in your life. There will be people that will mentor you along the way. So I didn't. So my background is actually, I am a civil engineer that instead of doing business classes, did biology because I like that too. So my undergraduate degree is, I shifted out at the end with a few classes to go in engineering, which I picked up later. So I have an engineering degree. I have a degree in botany, and I have a minor in applied mathematics and a minor in English literature. So I'm one of those really geeky people. I admit it. Although I did play field hockey, and I did play athletics on provincial and national teams. So I'm one of those really weird people that just really had a lot of interests and went pretty broad. But in my undergraduate, when I was finishing up as an engineer, I was in a small town in British Columbia working as at an internship at a mining company. And I realized really quickly that I was more interested in reclaiming the mine spoils than I was ever in actually mining. Engineering teaches you minimum effort for maximum profit, which has actually been a good lesson in my life. I don't regret the engineering at all. I think I solve problems better than a lot of other people because of it. My professor from the mycology class, which is the study of fungi, called my parents and asked where I was. And my mom said, well, she's, you know, out in central British Columbia. He actually phoned me and said, look, I have a visitor from Oxford, England, who's coming to give lectures and to meet students. Would you come in, take some days off and come in? So I took some days off and I came in. And he introduced me to Dr. Jack Harley. Dr. Jack Harley was the first person who wrote about mycorrhizas from Oxford University. He was elderly, but I listened to his seminar. And then my professor invited me out for dinner with them. And the next morning he asked me to come in and have a chat with Dr. Harley. Dr. Harley offered me a graduate position at Oxford. And when I got there, he died, unfortunately. And so I came back to the professor, Dr. Dennis Parkinson at the University of Calgary, where he had wanted, the whole idea I found out was that Dr. Harley was actually supposed to offer me graduate work with Dennis, not with him in Oxford, but I got the reverse offer, which was fine. It was wonderful to go there, and I would have stayed had I been able to do what I thought I wanted. So really, a lot of the reason I'm where I am is because of really amazing people that took it upon themselves to mentor me, to say, hey, you know, I think you have some really good ideas. Come and do graduate work. So I've been incredibly blessed that way. I've always been interested. You know, like I said, I won the science fair when I was in grade 8, and I always wanted to go to university. I'm also one of those weird kids. My birthday's in November, so I was always a lot younger than everybody else, and I started school a year early. So I went to university when I was 16. But I couldn't wait to go to university. Oh my gosh, it was just magical to get into that level of learning. And but also a real, you know, sometimes we think that our teachers are being a bit tough on us, but we have to remember, we get to university, the profs don't have time to mentor you at all unless you actually get into the higher level classes. So be prepared. It's all up to you. Build your habits now, because it's going to be important as you get to university, because nobody's going to care that you didn't hand in your homework. And they're not necessarily going to give you a break either. So, but yeah, pay attention. And if there's people whose work you really like that you come across, ask them questions. Be interested, engage, and you'll be surprised what comes out of it. Opportunity, lots of opportunity.
Awesome tips. Thank you so much, Dr. Clapperton. I think we should let you go. I think we're bumping right up against time here, and I know the students have other classes to get to as well. I would like to ask each of the schools to let me know how many students participated today, just so we can gauge the effectiveness of our event. Also, if you have any additional questions, you can reach out to me here at the NRD, or you can reach out to Dr. Clapperton. Dr. Clapperton, can you bring up that slide one more time with your contact information on it, so that
I can
jot that down? You can reach out to her via email and Twitter, I believe, were the two options she had out there. So we'd love for students to reach out to Dr. Clapperton with additional questions. As she mentioned, mentoring is just such a vital part of your career preparation. So definitely take this opportunity, students, to make that connection. With that, I just want to say thank you so much to Dr. Clapperton, and thank you to all of the student participants today.
Yeah, thank you all. Thanks for your attention today.
Thank you.
Thank you.
Bye-bye.
All right, stay well.